Medical research relies on rigorous experimental and clinical study design to ensure valid and reliable findings. Robust research protocols are essential for drawing accurate conclusions and informing clinical practice. This section will explore the key principles and methodologies that form the foundation of effective study design in the medical field. Understanding these core concepts is crucial for conducting high-quality, impactful medical research. We'll cover topics like control groups, randomization, blinding, and statistical analysis to highlight best practices in experimental and clinical study design. A well-designed research study is crucial to minimizing bias and ensuring the validity of the findings. Robust research design enables other scientists to replicate the study, building confidence in the results and advancing scientific knowledge. Appropriate sample sizes and statistical methods increase the ability to detect meaningful effects and draw reliable conclusions. Strong research design allows for the establishment of causal relationships between treatments and outcomes, rather than just correlations. These elements of rigorous research design are essential for producing high-quality, impactful scientific work. RCTs are considered the gold standard for evaluating medical interventions as they minimize bias and confounding factors. Participants are randomly assigned to either the treatment or control group, ensuring comparable groups for a fair comparison. The control group receives a placebo, allowing researchers to isolate the specific effects of the intervention. Participants, researchers, and/or clinicians may be blinded to the treatment assignment to minimize bias. Double blinding is a key feature of clinical studies, where neither the participants nor the researchers know which treatment each patient is receiving. This helps eliminate bias and ensures an objective evaluation of the intervention being tested. Placebos, or inert treatments, are used as a comparison to the active intervention. Blinding is crucial to ensure participants cannot distinguish the placebo from the real treatment. Blinded outcome assessment is essential to prevent researchers' expectations from influencing the evaluation of treatment effects. Blinded clinicians can objectively measure patient outcomes. Blinding at multiple levels, participants, researchers, and outcome assessors, is a powerful tool to enhance the validity and reliability of clinical study findings. The placebo effect is a fascinating phenomenon where patients' symptoms improve simply from the expectation of receiving treatment, even if the treatment itself has no inherent therapeutic value. Placebos are commonly used in clinical trials as a control to isolate the true effect of an experimental treatment. By comparing the treatment to an inert substance, researchers can determine if the observed effects are due to the treatment itself or just the patient's belief in receiving treatment. Researchers study the placebo effect to understand how patient beliefs and expectations can influence treatment outcomes. The goal is to harness this effect to enhance the therapeutic benefits of treatments. However, the use of placebos in clinical trials raises important ethical considerations, as patients may receive an ineffective treatment instead of the standard of care. This is an ongoing challenge that researchers must carefully navigate. Crossover studies allow each participant to act as their own control, reducing variability and increasing statistical power. Key features are the paired comparison, where each participant receives both the treatment and control interventions in random order. There is also a washout period between the two treatments to allow the effects of the first intervention to subside. We need to consider potential carryover effects, where the first treatment may influence the response to the second treatment. These design elements help us isolate the true treatment effect and account for individual differences between participants. Parallel group studies involve randomly assigning participants to different treatment groups that operate independently throughout the study. This allows for a direct comparison of the effects of the interventions being tested in each group. Parallel designs can be more efficient than crossover studies, as they avoid carryover effects and don't require a washout period. The independent nature of the groups and simultaneous comparison makes parallel designs a useful and effective study approach. Factorial studies allow us to evaluate multiple interventions or factors simultaneously, providing a more efficient and comprehensive assessment. These designs can uncover interactions between different factors, revealing how the combined effect of two or more variables differs from the individual effects. By incorporating multiple factors, factorial studies can increase the statistical power to detect significant effects, leading to more robust and reliable findings. Factorial designs allow for flexible data analysis, enabling us to investigate the main effects of individual factors as well as their interactions. The visual diagram on the slide illustrates a 2x2 factorial study design, clearly showing the different study arms and the concept of simultaneous evaluation. Observational studies allow us to closely observe and record behaviors, trends, and associations in the real world without actively intervening. While these studies can identify relationships between variables, they cannot definitively establish cause and effect like experimental designs can. However, observational approaches provide valuable insights into natural human behaviors and health outcomes in real-world settings. This gives us a deeper understanding of how things work in the real world, beyond what we can learn in a controlled laboratory setting. Case control studies look back in time to identify factors that may contribute to a specific outcome or condition. Researchers identify a group with the outcome, cases, and a group without the outcome, controls, for comparison. By examining past exposures, case control studies can help determine which factors are associated with the outcome. This retrospective design allows us to efficiently study rare outcomes and investigate multiple potential risk factors. The comparison between cases and controls helps us understand which exposures or characteristics are linked to the condition of interest. Identifying these risk factors is a crucial step in understanding the causes of a disease or outcome, which can inform prevention and treatment efforts. Cohort studies are a type of longitudinal research design. They follow a group of people over an extended period, collecting data and observing outcomes as they unfold. Researchers identify an exposure or risk factor, then observe how it relates to the development of a specific outcome or disease. This is an observational approach. Researchers do not intervene or randomize, but simply observe and analyze naturally occurring patterns. Cohort studies provide valuable insights into the relationships between exposures and outcomes over time. Meta-analyses and systematic reviews are powerful tools that help us synthesize evidence from multiple studies. Meta-analyses use quantitative statistical analysis to combine data, increasing statistical power and resolving uncertainties when individual studies disagree. They provide a numerical estimate of the overall effect size. Systematic reviews take a comprehensive and structured approach, following a predefined protocol to identify, evaluate, and summarize all relevant evidence on a specific question. They provide a qualitative synthesis of the available evidence. Both approaches give us a higher-level perspective and help us identify patterns, trends, and gaps in the research. Establishing causality is about identifying the relationships between different variables and determining if changes in one variable directly cause changes in another. This involves carefully designed experiments with control groups and statistical analysis to rule out alternative explanations and establish causal links. Rigorous experimental design, including randomization, is essential for establishing causal relationships. Statistical techniques like regression analysis can help quantify the strength and direction of relationships between variables. Understanding causality is crucial for making informed decisions and developing effective interventions. Confounding variables are factors that influence the relationship between the independent and dependent variables, but are not the focus of the study. If not properly controlled for, confounding variables can mask or exaggerate the true relationship between the variables of interest. Careful study design, background research, and statistical analysis are needed to identify and account for potential confounding variables. Effectively controlling for confounding variables is crucial for drawing valid conclusions and minimizing bias in medical research. Medical research can be susceptible to various forms of bias, including selection bias, measurement bias, and confirmation bias. These biases can distort the research findings and lead to misleading conclusions. It's important to be aware of these different types of bias and understand how they can impact the validity and reliability of medical studies. Strategies to minimize bias include randomization, blinding, and using validated measurement tools. Careful study design is crucial to mitigate the impact of bias. Publication bias and selective reporting of results can also distort the medical evidence base. Transparent and complete reporting of research methods and findings is essential to address this issue. Ultimately, Awareness and mitigation of bias in medical research is critical, as it can have significant implications for clinical decision-making and patient care. Randomization is a key strategy to minimize bias by ensuring systematic differences between treatment groups are eliminated. Blinding participants, researchers, and assessors helps prevent biases that can arise from expectations or preconceptions. Standardized procedures across the study ensure data is collected and analyzed objectively without introducing variability. Independent reviews by external committees can provide an unbiased assessment and identify potential sources of bias that the research team may have overlooked. Intention to treat (ITT) analysis is a crucial approach in clinical trials that helps preserve the benefits of randomization. ITT analyzes participants based on their original treatment assignment, regardless of adherence or protocol deviations. This helps minimize bias by avoiding the selective exclusion of participants, which can skew study results. ITT analysis typically provides more conservative estimates of treatment effects compared to per-protocol analysis, as it includes all randomized participants. The rigor and attention to detail in ITT analysis are essential for ensuring the validity and reliability of clinical trial findings. Per-protocol analysis includes only the participants who completed the study as intended, following the exact protocol, without deviations. This analysis provides insight into the efficacy of the intervention when it is delivered as intended, without the influence of noncompliance or dropout. However, per-protocol analysis can introduce bias by excluding participants who did not fully adhere to the protocol, potentially limiting the generalizability of the results. Adaptive clinical trial designs allow us to be flexible and make adjustments during the study based on emerging data. This flexibility can come in different forms. We can do interim analyses to evaluate the data at multiple checkpoints and guide our decisions. We can also reassess the sample size based on the interim results, increasing or decreasing the number of participants as needed. The goal is to optimize the trial and increase the chances of detecting a meaningful treatment effect. Adapting the study in real time based on the data gives us a better chance of success compared to a more rigid, traditional trial design. Interim analyses are a crucial part of clinical trials, allowing us to evaluate the data and make important decisions about the study's direction. The purpose of these analyses is to gain insights that can inform whether the trial should continue as planned, be modified, or even terminated. Timing is key. These analyses are scheduled at specific points, often based on the number of events or participants that have been observed so far. However, we must balance the need for these early insights with the risk of introducing bias or compromising the overall integrity of the study. Careful planning and execution of interim analyses is essential to ensure we make the best decisions for the trial and the participants. Bayesian clinical trial designs use probability models to quantify uncertainty and update beliefs as new data emerges. This probabilistic approach allows for more adaptable trials, where study parameters can be modified in response to interim results. Bayesian trials can also reach conclusions more quickly by continuously incorporating new evidence, leading to faster timelines. The key advantages are the ability to better quantify uncertainty, adapt the trial design, and accelerate the overall process. Ensuring participants fully understand the risks, benefits, and voluntary nature of the study is a fundamental ethical requirement for medical research. Independent institutional review boards, IRBs, play a critical role in reviewing research protocols to safeguard the rights and well-being of human subjects. Extra protections are needed for vulnerable populations, such as children or the elderly, who may have limited decision-making capacity. Researchers must carefully weigh the potential benefits of the study against any risks or burdens to participants, striving to minimize risks and maximize the ethical value of the research. The informed consent process is critical to ensure participants fully understand the study and their rights. We must clearly explain the study's purpose, procedures, risks, and potential benefits. Participation must be completely voluntary. Participants can withdraw at any time without penalty. We need to verify participants understand the information and have the opportunity to ask questions. Participants must provide written informed consent, which is documented and kept on file. Institutional review boards, or IRBs, are independent committees that carefully review and approve research proposals to ensure they protect the rights and welfare of human participants. IRBs assess the ethical considerations of studies, evaluating the risks, benefits, and informed consent procedures to uphold key ethical principles in medical research. Importantly, IRBs strive to consider the needs of diverse populations and ensure fair representation in clinical trials so that the research benefits all segments of society. IRBs play a critical role in the medical research process, providing essential oversight and approval to safeguard the well-being of study participants. Designing clinical trials for children requires special considerations around dosing, safety, and informed consent from parents/guardians. Older adults often have more complex medical histories and drug interactions, so we need to tailor inclusion criteria and outcome measures for geriatric trials. Ensuring adequate representation of minorities, women, and underserved groups is crucial for the generalizability of medical research findings. This helps ensure the results apply to the broader population. Overall, we need to thoughtfully design trials to be inclusive of special populations like pediatric and geriatric participants, as well as diverse racial, ethnic, and gender groups. This allows us to develop treatments that work effectively for all patients. Pediatric clinical trials require additional ethical considerations to protect vulnerable children. Informed consent from parents or guardians is crucial, and the trial's risks must be carefully weighed against potential benefits. Children have unique physiological and developmental differences from adults, necessitating specialized trial designs, dosing, and safety monitoring. Recruiting and retaining pediatric participants can be more challenging. Pediatric trials are subject to stringent regulatory requirements to ensure the protection of child participants. Institutional review boards and data safety monitoring boards play a critical role in oversight. Pediatric trials are especially important for rare diseases that predominantly affect children. Collaboration and data sharing among researchers are essential to advance treatments for these patient populations. Geriatric patients often have multiple health conditions and take various medications, so we need to carefully design the clinical trial to account for this complexity. As people age, their bodies change in ways that affect how drugs are absorbed, distributed, metabolized, and eliminated. We must consider these age-related physiological changes when determining appropriate dosing and evaluating safety. It's important to include measures of physical function, cognition, and quality of life in these trials to fully understand the impact on older adults beyond just the primary medical outcome. Older adults may face barriers to participating in clinical trials, such as transportation challenges or caregiver responsibilities. We'll need to use targeted recruitment strategies and engage with caregivers to overcome these barriers. Data and Safety Monitoring Boards, or DSMBs, provide independent oversight and monitoring of clinical trials. Their role is to ensure participant safety and data integrity throughout the trial. DSMBs regularly review unblind safety and efficacy data to identify any emerging concerns or issues that could impact the trial. They also provide ethical guidance, ensuring studies adhere to the highest standards and protect the well-being of research participants. This rigorous evaluation and oversight helps maintain the integrity of the clinical trial process. Clinical trials are crucial for advancing medical knowledge and improving patient outcomes. Registering trials in public databases like clinicaltrials.gov ensures transparency and accountability. Detailed reporting of trial methods, results, and data promotes scientific integrity and allows others to build on the findings. Timely disclosure of trial results, both positive and negative, benefits the entire medical community. Sharing information openly helps drive scientific progress and informs better decision-making for patients and clinicians. The key steps in translating research into clinical practice are: review findings, carefully analyze research results to determine their clinical relevance and applicability, pilot implementation, test new approaches in a small, controlled clinical setting to evaluate their effectiveness, broader adoption, scale successful interventions across healthcare systems and providers to drive wider impact. This process is crucial for advancing medical care by ensuring research findings can be effectively implemented in real-world clinical settings. The goal is to thoroughly vet new approaches, pilot them in a limited context, and then roll out the most promising interventions more broadly across the healthcare system. This slide brings our presentation to a close, highlighting the critical role of medical research in advancing our understanding of health and disease. Rigorous research design is essential for uncovering insights into the complexities of the human body and the factors that influence our well-being. By carefully designing and executing research studies, we can uncover new knowledge that leads to transformative advancements in patient care and treatment. The work we do in the lab has the potential to dramatically improve lives, which is why it is so important that we remain dedicated to this vital field of study.